Morning, people. I hope you're doing well, my lovelies, because what we've got here may contain science. Toxic science. And no, not toxic like academia drama. Toxic like the poison dart frog. They are wildly, excessively poisonous. Like incredibly overkill poisonous. Like allegedly enough poison to kill 20,000 mice or 20 full-grown humans. Poisonous. But until this study from Stanford researchers published in the journal eLife, I'd never really asked myself, how do poisonous dart frogs, whom these researchers describe as, quote, just a ball of toxins, avoid poisoning themselves? As soon as I asked that, though, I realized I don't even know how poison dart frogs got their poison in the first place, or what kind it was. Well, frogs aren't a venomous species like some snakes that synthesize their own toxins, building specialized danger proteins that attack neurons or blood vessels or blood cells. Dart frogs don't make their own poison. Instead, they are profiteers in a conflict that has been going on for millions of years. Plant-animal warfare. See, lots of plants don't want to be eaten. Sure, some do, making their fruit tasty and full of sugar so animals will eat them and propagate the plant's seeds, but other plants don't roll that way. Or they're okay with some parts being eaten, like that fruit, but not other parts that they need, like the roots. To discourage animals from eating them, then, lots of plants synthesize small molecules called alkaloids. Now the plants, they don't know animal biology. They're plants. So it's not like they can somehow custom design alkaloids to affect animals, but the plants that manage to synthesize alkaloids that biologically affect, you could say, poison animals, are more likely to not get eaten and, by implication, live to reproduce. That's evolution, baby! So, plants evolved to synthesize these alkaloids that poison animals. Problem is, animals still gotta eat plants. Life needs things to live, after all. And over time, animals that can break down these alkaloids are more likely to be able to eat poisonous plants, live through the experience, and go on to reproduce because that's evolution, baby! And so it has been over these millions of years. Evolutionary pressure for plants to develop ever more potent alkaloids to avoid getting eaten, animals evolving ever more sophisticated metabolism to disarm these poisons. It's one of the big jobs our liver does for us. And that's why so many of our pharmaceuticals come from plants. They started off as molecules that were selectively pressured to have a biological effect on animals. And while animal species have evolved metabolisms that break down a lot of these alkaloids, there are still plenty that poison the heck out of most animal species. And that's just what the poison dart frog uses. Their poison comes from plant alkaloids that poison the heck out of most animals. Now, poison dart frogs don't eat plants themselves, but they do eat insects that eat the alkaloid-containing plants because that's how the food chain works. Of course, at this point in the food chain, most species would use the typical plant-animal warfare playbook. Try to convert the poisonous molecule from the bug they ate into something non-toxic by chemically altering it, like adding an extra oxygen molecule or some similar cheap hack. But that's just not the poison dart frog's style. Instead, the dart frog accumulates these really aggressive alkaloids. And if a bird decides to get a bite of frog, that bird is getting a megadose of millions of years of plants' don't-eat-me evolution concentrated from the frog's own diet. And that brings us back to the first question. How do dart frogs avoid poisoning themselves? They can't convert the poisonous alkaloid into something non-toxic, after all. If you do that, you disarm the weapon. It won't harm you. But it also won't harm predators that want to eat you. So, the frog has to keep the alkaloid in its active form, keep the weapon armed, without being affected itself. So, the frog keeps the poisonous alkaloid as is, but binds the alkaloid to a safety protein, so the frog can remain unharmed. Fortunately, vertebrate animals already have the kind of protein our frog needs. They're called transport proteins, and we use them to bind all sorts of things. Albumin is the most common such transport protein, and it literally makes up half of all proteins in human blood plasma, and it binds all sorts of things, from small inorganic molecules like water, to pharmaceuticals, to fellow proteins like hormones. There are also transport proteins that specialize in particular diet-derived molecules, like there's a specific vitamin D binding protein. These transport proteins don't necessarily chemically affect the things they bind to, like albumin isn't metabolizing H2O into H2O2 or something, but instead, these transport proteins just attach to the target and stick to it as they move about the circulation together. That sounds like exactly what our poison dart frog needs, a protein that binds to the diet-derived alkaloid so that the alkaloid's active site can't interact with and do mischief to the dart frog's cells. And indeed, that's exactly what our Stanford researchers went in search of. Okay, great. Sounds like a plan. But how do you find one particular protein needle in an entire poison dart haystack? Apparently, you set a probe with a lure attached. Our scientists took a competitor of pumilotoxin, one of the alkaloids we know poison dart frogs accumulate, and attached a fluorescent chemical compound to it. If we've built our probe lure correctly, then it will bind to a protein in poison dart frog plasma, but it won't attach to anything in plasma from a non-poisonous frog species, because non-poisonous species don't bind to and accumulate the poison. They disarm it, get rid of it, or die trying. Once our scientists validated that the probe bound to the protein in poison frog plasma and not plasma from non-poisonous frogs, it was time to reel it in and see what we caught. So we repeat the experiment, only this time, instead of using a fluorophore to see that our lure bound to stuff, we instead use a biotin handle that we can easily grab back after we've put it into the frog plasma. Once we've done that, we can perform protein analysis and see what it bound to. We call the result alkaloid-binding globulin. After names like brain-aware and e-soil, alkaloid-binding globulin seems a little boring, like calling a radio telescope the Very Large Array because it's an array and it's very large. But at least I know exactly what this protein and that telescope are. Now, our researchers are a little surprised that the albumin we talked about before doesn't do the job in poison dart frogs. After all, we just got done saying that albumin is fantastic at binding to a whole diverse range of molecules. But if you ask me, an alkaloid that can be easily disarmed with the most common protein in animal plasma would be a real beginner-level poison, and the frog's predator could deactivate it just as easily as the frog. That's not a real deterrent. Instead, our poison dart frogs have this specialist protein that binds to a small number of different poisonous alkaloids which, surprise, surprise, correspond to the poisons our dart frogs accumulate. Indeed, different species of poison dart frog have slightly different variants of this alkaloid-binding protein, and we suspect that corresponds to the different alkaloids each frog species loads up on. And when we stained for this alkaloid-binding protein in frog tissue, we found most of it in the intestines, where the frog's first going to encounter the alkaloid, a ton in the liver, which is the big toxin management organ of the body, and in the skin glands, where poison dart frogs keep their accumulated poison and secrete it onto their skin. And that is exactly why frog biologists do not lick the science. At this point in the paper, our scientists ride off into the sunset, having discovered how poison dart frogs don't poison themselves. But for a moment it left me wondering, okay, that's how poison dart frogs don't poison themselves. But the whole point of this specialist alkaloid-binding globulin is that the alkaloid itself could be kept active as a deterrent. How can the poisonous alkaloid be a deterrent if there's a safety protein attached? Do they cleave the protein from the alkaloid when it's secreted onto the skin? Why aren't these researchers talking about it? Oh. Although this isn't specifically covered in the paper, although there could be another way the dart frog does this, there is at least one clever little mechanism that would let the little froggo make sure they don't die from their own supply, but maintain the threat for any predators that decide, I can eat the brightly colored thing, I'm built different. How? Because we're dealing with a small molecule bound to a protein. And digestive systems interact with each of these very differently. Not all, but plenty of small molecules make it through the digestive system without breaking down. After all, if our stomach acid destroyed vitamins and minerals, for instance, we'd be screwed. So the alkaloid makes it through the stomach. Again, any plant-derived alkaloid that didn't make it through an animal's stomach wouldn't have been an effective deterrent for the plant either. So evolutionary pressure has selected toward producing alkaloids that survive the stomach's low pH. But while many small molecules like our poisonous alkaloid leave the stomach in one piece, proteins don't. Between acid that denatures the protein and enzymes that break them down, animal stomachs are built to digest proteins into their fundamental components so those amino acids can be recycled and reused. And when a bird or other predator makes its last mistake and eats that froggo, its stomach typically digests the alkaloid binding protein, removing the safety that kept the dart frog from poisoning itself and rearming the poisonous alkaloid once again. Bye-bye, birdie. So to recap, the poisonous dart frog eats bugs that ate poisonous plants. Most species either avoid the plant entirely or they metabolize its poisonous alkaloid into a deactivated form. But dart frogs instead have a specialized protein that can bind to some types of these poisons. By binding a protein to the active site of the poisonous molecule, the frog is protected from the poison it accumulates. If a predator eats that frog, though, the predator's stomach will digest the protein, but not the poison, leaving the still active molecule to wreak toxic havoc on the predator that dared chomp a nom-nom on our little froggo. Altogether, I don't blame predatory species for simply avoiding the dart frog and eating other frog species. Indeed, most species that do prey on the poison dart frog, such as the damselfly nymph, prey on the tadpoles rather than the adults. The only species that does go in against this amphibian when death is on the line is the fire-bellied snake, which preys on one species of dart frog and can seemingly only do so due to a mutation that affects cells throughout their nervous system, muscle, and heart, but also prevents them from being affected by that dart frog's most lethal poison. Personally, I'm not sure I would take the fire-bellied snake's trade-off. The dart frog's adaptation gives them a brilliant defense against predators, and we finally understand a little more about how they do that. With any luck, understanding alkaloid-binding globulin will help us develop better antitoxins to the alkaloids that currently knock us flat. And maybe prevent predators from eating us. I'm told being pretty colors helps.